Welcome once again to Bio 211D, Plant Physiology, Unit 5, Session 4, Transport of Organic Solute. And by the close of this session, you should be able to explain translocation, describe the structure of tissues involved in translocation, show the direction and pathways of translocation, list the organic solute translocated, describe the patterns of translocation, source to sink, and explain sugar loading and unloading. Translocation, the movement of organic foods such as sucrose and amino acids in phloem from regions of production to regions of storage or regions of utilization in respiration or growth. So plants synthesize food materials by photosynthesis and store them in the chloroplast found in mesophyll cells. The site of synthesis and the site of need are separated by time and space. The structures found between these sites play a significant role in translocation. We have already established the fact that the phloem is the tissue that translocates the products of photosynthesis from mature leaves to areas of growth and storage, including the roots. As we will see, the phloem also redistributes water and various compounds throughout the plant body. In order to gain a clearer understanding of the phloem, it is necessary to integrate the structure of the phloem with its function. The phloem consists of three different cells. These are sieve elements, companion cell, and intermediary cells. You should also note that in some cases, the phloem tissue also includes fibers and sclerates and lacicifers. However, our attention will be focused on the latter three groups of cells which are involved in the transport of organic solutes. The sieve elements, they join end to end to form the sieve tube. They also represent the most active food conducting elements in the phloem tissue. The sieve tubes are found arranged parallel to one another from one end of the plant body to another. Each sieve tube is formed by a series of hollow cylindrical cells called sieve tube cells arranged one above the other. So the sieve tube cells are separated from each other by horizontal perforated plates called sieve plates. The sieve cells communicate with each other through the sieve plates. Each sieve cell has a thin cell wall which is composed of only cellulose. The cell has a central mass of dense centroplasm. The granular cytoplasm forms a number of projections called cytoplasmic strands extending towards the sieve plate. The nucleus is absent. Companion cells, they are more or less spindle-shaped cells associated with the sieve tube cells. Each companion cell is found attached to any one lateral surface of a sieve cell. The companion cell and the neighboring cell, sieve cell, together represent a pair of sister cells. The companion cell has a granular cytoplasm, prominent nucleus, and one or two small vacuoles. The nucleus of the companion cell is said to be capable of exerting its influence on the adjacent sieve cells. They perform some of the basic cell functions for the sieve tube members like protein synthesis. They have numerous plasmodesmata connecting them to surrounding cells, particularly to the bundle sheath cells. Intermediary cells also have numerous small vacuoles, as well as poorly developed thylakoids and a lack of starch grains in the chloroplast. So you have a picture of that being shown. You have the parenchyma cell, you have the sieve tube cell, companion cell, and plasmodesmata as indicated in the plant. The stem, we have the xylem, the phloem, you can have the vascular bundle, the vascular tissue, you have the gland tissue, the derma epidermis, right, as indicated, a section through the stem. So Over here we have the end wall companion cell and the sieve tube, how it is readily displayed in the plan. Intermediary cells, schematic drawings of mature sieve elements or sieve tube elements. You have the external view showing sieve plates in lateral view areas. Longitudinal section showing two sieve tube elements joined to form a sieve tube. The pores in the sieve plates between the sieve tube elements are open channels for transport through the sieve tube, as we just saw in the past slide. The plasma membrane of a sieve tube element is continuous with that of its neighboring sieve tube element. So intermediary cells, each sieve tube element is associated with one or more companion cells which take over some of the essential metabolic functions that are reduced or lost during differentiation of the sieve tube element. Note that the companion cell has many cytoplasmic organelles, whereas the sieve tube element has relatively few organelles. The source is by Theis and Zager in 2006. So, direction and pathways of translocation of organic solids. The two long-distance transport pathways, the phloem and xylem, extend throughout the plant body, and the phloem is generally found on the outer side of both primary and secondary vascular tissues. So you have the root and stem structure of a plant, and in there you see the epidermis being the outer, then in that we have the cortex, then the vascular bundle, where we have the phloem to the outer and xylem to the innermost part. So directions of translocation. Translocation of organic solids may take place in the following directions. Downward translocation. Mostly the organic material is manufactured by leaves and translocated downward to stem and roots for consumption and storage. Upward translocation. It takes place mainly during the germination of seeds or tubers, etc. When stored food, after being converted into soluble form, is supplied to the upper growing part of the young seedling till it has developed green leaves. Upward translocation of solute also takes place through stem to young leaves, buds, and flowers, which are situated at the tip of the branch. Lateral translocation, radical translocation of organic solutes also take place in plants from the cells of the pit to cortex. Intermediary cells, part of the translocation of organic solute. Part of downward translocation of the organic solute takes place through the phloem, and this can be proved by the ringing experiment. Part of upward translocation, although translocation of organic solutes take place through the phloem, but under certain conditions it may take place through the xylem. Part of lateral translocation from pit to cortex takes place through medullary rays. Organic solute translocated. Water is the most abundant substance transported in the phloem. Dissolved in the water are translocated solids, mainly carbohydrates. Sucrose is the sugar most commonly transported in seeded meats. Nitrogen is found in the phloem, largely in amino acids and amides, especially glutamate and aspartate, and their respective amides, glutamine and asparagine. Almost all the endogenous plant hormones, including auxin, gibberellin, cytokinins, and abscisic acid, have been found in the sieve elements. Patterns of translocation. Translocated materials always move from areas of supply, called sources, to areas of metabolism or storage, called sinks. A source includes any exporting organs, typically mature leaves that are capable of producing photosynthate in excess of their own needs. The term photosynthate refers to products of photosynthesis. Another type of source is a storage organ during the exporting phase of its development. A sink includes any non-photosynthetic organs of the plant and organs that do not produce enough photosynthetic products to support their own growth or storage needs. Roots, tubers, developing fruits, and immature leaves. So you have the xylem and you have the phloem. One way only. That's the movement of minerals and ions. So water and minerals. No end walls between cells. You have thick walls with different length. So it is one way only, moves in only one way. But in the phloem, you have two-way flow. Whilst one is moving up, another is moving down. Water and food particles are displaced, and cells have end walls with perforations, but that doesn't occur in the xylem vessel. So sugar loading and unloading. There are two known mechanisms operating in different species. In one, sucrose enters the cell walls near the phloem in the smallest veins of the leaf. It then enters the phloem by attaching to sucrose transporter proteins embedded in the plasma membranes of the sieve elements and companion cells. In the second mechanism, sucrose enters the companion cells of the minor vein through small plasmodesmata and is converted to larger sugars, raffinose and stachyls. These larger sugars are unable to diffuse back through these plasmodesmata due to their size. Therefore, they are trapped in the phloem of the leaf and build up to high concentration. They enter the sieve elements through larger plasmodesmata and are carried away toward the sinks. When sugars and other nutrients arrive in sink tissues, they unload from the phloem and enter surrounding cells, either through plasmodesmata or by crossing from one cell to another across the cell walls. The size and metabolic activity of the different sinks determines the amount of material that is delivered to them. Thus, the use of sugar in the sinks determine how much sugar flows to them. So in review, translocation in the phloem is the movement of the product of photosynthesis from mature leaves to areas of growth and storage. The phloem also redistributes water and various compounds throughout the plant body. Aspect of phloem translocation. The pathway of translocation, sugars and other organic materials are conducted throughout the plant in the phloem, specifically in cells called sieve elements. And the sieve element display a variety of structural adaptations that make them well suited for transport. Patterns of translocation. Materials are translocated in the phloem from sources to sinks. Sources are usually mature leaves. Sinks include organs such as roots and immature leaves and fruits. Materials translocated in the phloem. The translocated solutes are mainly carbohydrates, and sucrose is the most commonly translocated sugar. Phloem sap also contains other organic molecules such as amino acid, proteins, and plant hormones such as well as inorganic ions. Rates of movement in the phloem are quite rapid, well in excess of rates of diffusion. Phloem loading and unloading. Transport of sugars into and out of the sieve elements is called sieve element loading and unloading respectively. Mechanism of translocation. Pressure mass flow is well accepted mechanism of phloem translocation. In this model, the bulk flow of phloem sap occurs in response to an osmotically generated pressure gradient.